And good morning, molecular genetics fans. So, I have a guest lecturer here in Quinkle. You can hear her tinkling there, scratching herself. She's here to talk about the dog genome and phenoplasticity, but she gets shy. Now, today we're actually getting into chapter 5, and just so you know, I got sort of a take-home thing that's going to cover a variety of chapters. Probably another week or so. I'm not sure how much further I need to go into it, but that's going along. And we're going to start getting into a bit more of a PCR-based experimental design, sequence analysis. That'll be a bit more free-form. We'll see how that goes. And that's what we have to say about that. So one of the points that is important in terms of modern molecular genetics is the organization of the genome, rather than just cloning genes, actually looking at the genomes in totality and how they evolve and how they're similar and different, and a variety of different kinds of issues surrounding what's in the nucleus. And one of the things that we're going to be addressing today is something called the N paradox. And N is the sort of abbreviation for the genome size, the number of bases in a genome. And just to sort of set it up, the paradox is this. When we begin looking at genome sizes, there are a range of genome sizes within different families of organisms, these phylogenetic families, and they can be dramatically different. And you would like to think that the genome size is going to be associated with complexity. With increasing complexity, you have an increasing genome size. But it doesn't seem to work that way. As we'll see, either the genome size is not going to be a direct correlation to complexity, or there's something about lilies that we don't know about. Lilies have a huge amount of DNA. In fact, a lot of plants have very large genomes. For that matter, the hermit crab, I used to work on the hermit crab doing some cloning, has a genome size the same as a human. Now, flies have a genome size that is a solid order of magnitude smaller. So there is ways of looking at this that make so you can go from simple organisms to more complex organisms. But there are some interesting things going on here. And so what we're going to begin doing is introducing what's in the genome organization. And our ultimate goal is to kind of develop how we can explain the N paradox. And we've got a variety of things we'll talk about with this. So a couple of terms. First is we have the genome, which represents the complete set of sequences in the genetic material. So when you're talking about cloning an organism's genome, you're basically talking about, or sequencing it, you're talking about actually identifying the sequence of every single nucleotide in that organism. And we're going to actually begin fitting in some DNA sequence technologies and things in this as well. So it is the sequence of every chromosome in the organism. Now, another kind of ome is something known as the transcriptome. And the transcriptome is the complete set of RNAs that are present in a cell or in a tissue or an organism. This is primarily messenger RNAs, but it also includes a variety of non-coding RNAs. And so the transcriptome represents the information that can be transcribed from a genome. So the next level up is something known as the proteome. And really, with each one of these things, we do increase in complexity, because the transcriptome represents the potential for intron-exon splicing, and so for various RNA splice isoforms. Now we have the proteome, which represents the complete set of proteins that is expressed by the entire genome. And the proteome is going to be different than the transcriptome because there's going to be post-translational processing. So we've got all kinds of things here. Now, a really critical ohm that is actually probably the last true frontier in terms of understanding the way these molecules interact, the way systems work, is something that is known as the interactome. Everybody has to have their ohm. The interactome is the complete set of protein complexes or protein-protein interactions that are present. And if you recall, when we have talked about protein structure, particularly in molecular cell, we talk about the primary structure of a protein, which is the amino acid sequence. And that can be inferred from the genome. And maybe you could say, okay, more accurately from the transcriptome, but regardless, you can deduce it from the genome. The secondary structure results in the interaction of neighboring R groups, and that can be fairly well predicted. The tertiary structure is non-neighboring R groups, and we can actually generate models that will predict that. This will be probably more than one specific shape, but still we can generate this. What we can't predict from the sequence is what is known as the quaternary structure. And I just sort of, out of a sense of indignation, I refuse to put quaternary structure into that discussion because you can't determine quaternary structure from the genome. Quaternary structure is protein-protein interaction. And that's probably the most critical thing to think about in terms of what a protein's doing, is what are the neighboring partners it's interacting with. And that's not going to be determined by genomics. That's determined by protein interaction, which is a much more delicate and difficult thing to screen for and to identify. But be that as it may, it is the next frontier. So another way of thinking about genomes is how we can map them. And we've talked a little bit about this already. We can develop what's called a linkage map. And just for the heck of it, I'll write this up there. Good vocabulary terms. So the linkage map was initially using mutations and developing first complementation groups, and then having multiple mutations on one chromosome, and then looking at recombination. And so you can tell how close mutations are, or these genes are to each other, by how frequent the recombination occurs. The further apart they are, the more frequent the recombination. The closer they are, the less frequent the recombination. Linkage maps have the drawback in that not all sites in the genome recombine at the same rate, but you can develop this linkage map. Another map that is possible is something called a restriction map. And in a restriction map, you're looking at the physical distances, the actual nucleotide distances, between restriction sites. It's another way of mapping things. The molecular characterizations of mutations and genes actually is used to reconcile and bring together linkage maps with the physical restriction map. And so, as we have mentioned before, because you have hotspots, the linkage map is somewhat distorted. The physical map is a way of actually putting genes into specific spots on the chromosome. And you can do this by sequencing an entire organism's genome, or the way the fly biz worked is for probably 20 years, people were doing old-fashioned gene cloning, mapping them onto polytene chromosomes and building up the genome. The fly genome was the first genome to be sequenced of a eukaryote. And one of the points that was made at a fly meeting that I attended back in the early 90s, or mid-90s, was the fact that the fly community had begun sequencing the genome to the tune of about 20% of the genome had been sequenced the old-fashioned way by looking at individual genes and identifying interesting genes and sequencing them first. So with the fly genome being a little bit smaller, that's possible. The human genome required a lot more strategy, and we'll be talking about that here in a bit. All right, what I'm doing here is I got slides and things, and I may throw them up, but I kind of like having the board. So one of the things we know that is a basis of evolutionary biology is that individual genomes can show a degree of variation. And that degree of variation is going to actually determine whether two organisms are within the same species or not. So we have the term we've talked about, what's called a polymorphism. And this actually brings about an interesting kind of definition of talking about mutation. Is that, if a gene has a sequence difference, is it a mutant? It is going to carry a polymorphism. I guess we would probably reserve the term mutation for something that actually has some kind of visible or identifiable effect. But we've got all kinds of changes that don't result in any kind of identifiable effect, and these are polymorphisms. So they can be everything from changes in the introns, that's easy. You can imagine that you can accumulate mutations within introns to a fairly high degree and not have any effect in the population. In molecular cell, if you recall, we were talking about alu inserts into introns, and they're all over the genome. We'll be getting into a little bit later micro-repeat sequences, things that the CODIS DNA sequence system is based on. Additionally, because of the way the nucleotide, the codon identification system works, the third nucleotide in a codon is often not required to be specific for identifying a specific amino acid. And so you can have a whole range of third nucleotide changes that don't change the amino acid composition of a protein at all. You can also have a whole range of changes that result in a conservative mutation, where you have one aliphatic amino acid substitute for another one, or one negatively charged amino acid substitute for another one. And so these conservative mutations can occur. And within a population, you can see these things. So the polymorphism levels at the sequence level can be very high, but generally they're not going to affect the protein's function, and they may not even be visible at the amino acid level. So one of the things that, as molecular techniques started to come about in the 80s, this idea of the linkage map wasn't limited to mutations, but it actually began to be expanded to polymorphic markers. And so something that plant breeders began doing before they had identified genes that were associated with specific traits, they would be able to identify a sequence polymorphism that was associated with the trait. And it wouldn't necessarily even be in the gene. It would just be very tightly linked. And they could then carry out linkage and recombination studies using that marker, not the gene, but the marker, the restriction site, and begin to select different breeds and things as they were doing as they're breeding plants for these specific kinds of traits. So there's a lot of stuff that has kind of moved forward. Now that we've got the entire human genome sequenced, and even more importantly, we're getting to a spot where we can sequence genomes very inexpensively, we're beginning to identify a whole range of what are really important, the single nucleotide polymorphisms. And these, of course, are probably the most important thing to think about when we start talking about using molecular genetics to identify mutations. Single nucleotide polymorphisms. A single nucleotide polymorphism, known as a SNP or a SNP, is, just as the name implies, a variation in sequence between individuals caused by a single nucleotide change. Now, as we've been saying, most of the time you can pick up single nucleotide mutations and not show any difference between two individuals. But there are occasions where you have a single nucleotide polymorphism that results in a stop codon being introduced into a critical gene. And it turns out that SNPs are pretty much responsible for most of the genetic variation between individuals, and the range of phenotypes that we see within the population are SNPs. The difference between a Dachshund and a Great Dane is all going to be a handful of single nucleotide polymorphisms. So single nucleotide polymorphisms, or SNPs, while they are the most critical variations, they are also some of the most difficult to actually identify, because you're now not talking about a change in a large amount of DNA. You're talking about a single nucleotide. In the beginning, single nucleotide polymorphisms were studied based on the potential for an SNP to change a restriction site. And this led to a whole sort of analysis and genome analysis that is known as, wait for it, restriction fragment, that's a G there, fragment length polymorphism, known in the business as an RFLP, or if you're particularly hip, it's referred to as a RIFLIP. And restriction fragment length polymorphisms are single nucleotide polymorphisms that happen to land in a restriction site. And so you can either generate a new restriction site or you can lose a restriction site. And one of the things that back in the 80s now, God, how many years ago was that? 40 years ago? Researchers were looking to identify DNA sequences that could be used as hybridization probes that would span across a site that was in the population a restriction fragment length polymorphism change. That is, you have a restriction site that would give you a certain number of fragments, and if that was lost, that would change the fragment size. And let's see, I think we have, a nice image that I'm going to show. My audience is coughing up along. You'll have to excuse them. Hey, Queen, come here. Come here. Oh, she's got to scratch herself. Come here. Can you say hi? Quinky wants to say hi. Don't be shy. Quinkle. She's not interested in restriction fragment length polymorphisms, I don't think. All right. So this is a blot that is showing RFLPs. All right. So we've got a DNA, a region of the genome that we have established a probe with. If we have a chunk of DNA, we can use it as a probe in a DNA hybridization experiment. And what we have then is this piece of DNA is spanning a region that has mutations in the restriction sites. And so we end up having DNA fragments that hybridize the probe of different sizes depending on whether the fragments are there. And so we have this slow-moving, a large allele, a smaller allele, and then two more, allele C and allele D. Now, we don't necessarily know what the effects of these alleles are. We simply know that we can identify them. And what researchers would do is exhaustively look for, kind of randomly, sequences around a gene that would actually show a restriction fragment length polymorphism associated with a mutation. And then you can actually begin tracking the mutation. Rather than actually tracking the gene, you're tracking a SNP that is very tightly linked. So we can use RIFLIPs and SNPs to actually begin developing very basic and rather primitive linkage maps. So this is a way to begin looking at the genome. Just as a little note here, the idea of DNA fingerprinting is a euphemism because it's not a fingerprint. It is a unique DNA sequence that is associated with individuals. And this is using potentially single nucleotide polymorphisms. These days, there are repeat sequences that are much more variable within the genomes in the population, and they're the ones that are really actually being used now. All right, so a really interesting experiment that used to be done a lot back in the 80s is the following. And it led to a really critical discovery. Whoop. And it is the following idea. It is something called a hybridization kinetics experiment. Sorry for all the jinking and jiggling around. A hybridization experiment in which you're doing an experiment like this. You have double-stranded DNA, hydrogen bonded. You boil it up. You make it single-stranded. And now you can analyze how much of the DNA is single-stranded. Now, obviously, at this point, when you boil it up, it is 100% single-stranded. So what would be done to carry out a kinetics reassociation experiment? So we're looking at what's called a reassociation kinetics. There's no K there, it's just C, regardless. A reassociation kinetics experiment. You're going to generate single-stranded DNA, and when you're doing this, what you typically do is fragment the DNA, generally by either passing it between two syringes, that'll do it, or exposing it to a sonicator with ultrasonic frequencies, and you sort of froth it up like you do your milk at a cappuccino machine, and you can break the DNA into small pieces, these small fragments, and then you denature it. And there are a variety of different calculations that we're not going to really roll into too hard. I'm actually going to post a document you might find interesting, you might not. You can use the concentration of the DNA and the time that it takes to reassociate and develop that into something that is known as the COT 1/2, or what's known in the business as a COT curve. And it goes like this. A couple of things to note. You can either by UV spectrophotometry tell whether DNA is double-stranded or single-stranded, or another standard way to do this is to pass the DNA over a calcium hydroxyapatite column. And calcium hydroxyapatite is actually part of the mineral that's in bone. Take ground up bones from somebody, you build a little column of it, and calcium hydroxyapatite, I think, it binds very, very tightly to single-strand DNA. And then you can begin by increasing the temperature, you can melt it off. So you take your solution, you pass it over, and you look to see how much comes out. And that's a measure of how much of the DNA has reassociated. So it's like this in terms of the tool. If you've got a genome that you can, sort of a hypothetical genome that is all A and all T. So you have A on one strand and T on the other strand. When you fractionate that up, break it apart, or denature it, any piece can hybridize with any other piece anywhere up and down along that chain. So you end up with a hybridization curve that will look like this. So all right, we're going to have the percent double-stranded. And we'll say 100% and 0. Now, just as I recall in one of the documents I've got, they may flip this around. However you want to do it, the information is going to be the same. It's just the axis is going to be, the curve may look a little bit different. So when you begin the curve, the experiment, you boil it up. All the DNA is single-stranded. You pass it through the hydroxyapatite column. All of it binds. You then allow this stuff to begin to renature. And so you start the experiment by boiling. You denature, and you start your stopwatch, or you stop your start watch. And you start measuring every 5 minutes or so the amount of double-stranded DNA. When you do that with a poly-A, poly-T, you see something that's going to look like this. So this would be just A base pairing with T. And that this is going to be effectively time, but because the DNA concentration is part of it as well, this is the COP 1/2 on this side, on this axis. It is more or less the time that it takes to renature, but because the concentration can be a part of this as well, you have to factor that in. Now, this is done under sort of standard saline conditions, standard salt conditions to allow for renaturing. All right, so you get this kind of curve. Now, when you begin looking at, say, a 10,000 nucleotide virus, a 10 kb virus, we see this. We see that the COT 1/2 curve is shifted down the x-axis because it's taking longer for these pieces to find each other, because now we don't have just a simple AT. We actually have unique sequence, and they have to actually line up, and they have to, in fact, find each other. What we can begin to identify is the overall size of a genome and its complexity can be analyzed by measuring its COT 1/2 and comparing it to like a known standard. And so this was the very first way that genome sizes were being analyzed, is by looking at the time it took for the genomes to begin to reanneal, to renature. So this renaturation kinetics. And so what we began to see is then you take a standard prokaryote that you have floating around the lab, a little bit of E. coli, and you see something that looks like this. It takes longer for it to actually hybridize completely, to reach 100% hybridization, because of the complexity of the genome, the size of the genome. And this is something that back in the 80s, folks were doing, running COT curves. And every time you identify a new organism or you find a new bit of material to grind up and extract DNA, you do a COT curve. Now, interestingly, with prokaryotes, you see a single kind of inflection point, a single line. With viruses, it's the same thing, shifted towards the y-axis here, it shifted down this way because it's a smaller genome. Prokaryotes, though, always showed this kind of relationship. When you start looking at complex eukaryotes, some interesting things start to happen. When you look at humans, for instance, you see something that looks like this. You actually have multiple bumps in the hybridization kinetics. And what this is taken to mean is that there are regions within the genome that exhibit different overall complexity. So the simpler the sequence is, the less complex it is, the faster it hybridizes. So you can imagine if you've got as it turns out, a repeat sequence that is 15 nucleotides long, and you've got a whole bunch of it, you actually can have that DNA sequence hybridized with any other spot. It doesn't have to hybridize as exact partner. It can hybridize up and down that repeat sequence. And so that's going to anneal faster than this stuff, and then this stuff. So one of the first things that was done was taking RNA and now radioactively labeling the RNA from a cell and throwing that into this mix and asking, where is the radioactive RNA hybridizing? So messenger RNA ends up hybridizing here. So this is where the mRNA is. And what falls out from this is we can actually begin to think of the genome in different degrees of complexity like this. We have this region that's represented by this first inflection is, in fact, a series of repeats. It is highly repetitive DNA. This sequence is moderately repetitive. And as work continued, what turns out is this is small sequence that's repeated over and over and over, and it ends up with a variety of different sequences there are referred to as satellites and microsatellites that are present in particularly mammalian and complex genomes at a huge amount. It makes up of the bulk of the genome content in a human. And it is not particularly useful or functional DNA. This sequence, the moderately repetitive, represents things that have repeat sequences, but the piece of DNA that is repeated is pretty big, say 1 or 2 kb. And an excellent example of moderately repetitive DNA would be things like the ribosomal RNA genes. So if you recall, the nucleolus in a eukaryote is made up of the RNA bubble, the ribosomal RNA bubble, that is being produced by the ribosomal RNA genes at such a high level that it actually creates this little discontinuity in the nucleus. And ribosomal RNA genes are fairly big, 2 kb or so, but they are repeated over and over and over again thousands of times. So this is a way of thinking about this middle section. It is repetitive, but it's a fairly long, complex sequence. And then we've got up here what is essentially unique single copy sequence. And that would be the actual RNAs, or the genes that code for messenger RNAs. So we have these 3 layers of complexity within a genome that are being identified by the use of Cott curves. So let's just look here at a figure that is showing the proportions of different sequence components in eukaryotic genomes. And we're going to begin to start to figure out this end paradox. Turns out, this is a little spoiler here, that what makes genomes dramatically different in size is going to be the amount of repetitive DNA they have. So, the take-home point here is the following. Thank you. In prokaryotes, there is virtually no repetitive DNA, in that it is all single copy, non-repetitive DNA, which means that in E. coli, as we were looking at with the Cott curve, it's going to hybridize at a single inflection point. The nematode C. elegans is a good model system. It's got a genome size now that's like between 10 to the 7th, 10 to the 8th nucleotides. A solid order of magnitude, almost 2 orders of magnitude bigger than E. coli. And it shows a fairly high concentration of non-repetitive DNA, and then we've got moderately repetitive, and then a very small portion of highly repetitive DNA. The fruit fly is a bit more complicated than the nematode. They've got even greater amounts of highly repetitive DNA. Check out mice here. They've got a even higher proportion, or at least a higher amount, but I guess proportionally different, of the highly repetitive DNA. But we begin to see the same sort of differences, that we have an increase in the amount of single copy DNA, non-repetitive DNA, which is owing to the fact that going from an invertebrate to a vertebrate mammal represents a pretty large increase in the amount of genes required to achieve that complexity. So here's Xenopus, and they've got a fairly large amount of DNA. Their genome size is pretty high as well, being vertebrates. They have even more moderately repetitive DNA. And then check this out. This is the plant, the basic tobacco plant, a standard dicot, and they have a larger genome size than mice or frogs, and primarily that's made up of repetitive DNA. So this is how we begin to think about genome complexities and genome sizes. Now, a large part of this repetitive sequence is what we've said is being fairly small sequences. It's made up of what are known as transposons. These are small DNA sequences that over evolutionary time have been jumping around. We're actually going to get into transposons and retroviruses in another week or so. But these are pieces of DNA that actually have the ability to replicate and move around the genome. As it turns out in the human population right now, we don't have any active transposons, but other organisms do. So the last little bit, and then we'll pick this up on next week, we can think about the identification of these unique sequences, because these are primarily protein coding genes, and they're going to be very conservative across a wide range of species. So you can begin looking at the genomes and you see all this repetitive DNA, but the pieces that are probably critical are going to be the pieces that are most conserved. And is there anything there fun to talk about? We will stop at this spot. Yeah. All right. So that's where we're going to stop. I'm going to be working on some sequence that I'm going to hand off to you for developing PCR primers that we're actually going to use, and we'll see how that works. Now that you guys are PCR primer experts, sure, why not? All right, that's all I got to say. I'm Dr. G. Peace.